Science of Cannabinoid Cultivation Academy. This is part 2 of a 5-part series. First one was back in November, and we're going to have another one going on pretty much every month until April. So I'm Daniel Enking. I'm the CEO of Everflux Technologies. We've got Brian Waxman here as well, and Dr. James White is our first speaker this morning, and I'll be passing it off to him in a minute. Just want to give a little quick introduction and make a few announcements. So just want everyone to know this event is hosted by Rutgers-University, which is a collaboration between Everflux Technologies and Leighton Morrison. Leighton Morrison, he's really kind of the brains behind this whole thing. He's, as you may know, he did a conference a couple of years ago about growing with regenerative and organic methods, and so we are continuing on that path here. We didn't use regenerative organic in the name because we really think, really what we're focused on here is just the science of how to grow plants and how to grow cannabis, and this is cutting edge science that has been coming to us in the last few decades. People like Dr. White have been uncovering how plants actually grow, and not really with fertilizers, but with microbes, with the help of microbes. So just a little bit about Everflux Technologies. What we do, we actually make microbial products, but we also really see ourselves as a platform for advancing regenerative agriculture and growing with living soil. So we didn't just want to make products that people are gonna add into their fertilizer regime, but we actually want to encourage people who might be growing with salts, with chemicals, to make a wholesale conversion over to growing with regenerative and organic methods. And so we make products that can enable that. We also make a inoculated biochar, where we put the microbes into the biochar so that's all ready to go in the soil and add to your organic matter. So that's what we're all about, and we started doing these educational programs, again, because we really just want to help people grow in a different way and be better at growing this way. So just want to quickly mention that our sponsors for this event, we have a title sponsor for the whole event, Agro Cocoa. They make cocoa coir products. Now, Leighton Morrison is really particular about the types of products that he works with. He goes to great lengths to kind of source and look at the quality with different products. And so, he was looking. We know that there's a lot of cocoa coir products out there that don't have the right pH and have kind of bad chemicals used in the process of making them and stuff like that. And so Leighton found Agro Cocoa and saw that they have a really, really good, high quality product, without harmful chemicals. So we only allow companies that we feel have really good products to sponsor this event. And so that's where Agro Cocoa comes in. And then our sponsor for this session is Pitmoss. If you're at the last event, you'll recall Pitmoss makes a substitute for peat moss. They make it out of recycled paper. And so peat moss just a couple kind of logistical announcements here. So if you were here last time, you recall that we had a chat, and we had different chat Q&A sections. This time we just have the one chat room. So you can post your questions there throughout. Brian and I will be keeping an eye on it, and when we get to the Q&A session, we'll try to bring those questions forward. You may want to reserve your questions for closer to when we get to the Q&A session, just to make sure that we get your question. Now, with Dr. White's presentation. We're actually going to stop a couple of times throughout the presentation, to answer some questions. He's kind of divided into different sections. So we'll let you know when we get to that, and then we'll start answering questions. So I think that's all the announcements. So with that, I will pass it off to Dr. White, who is really a leading expert in the subject of endophytes, and he's going to do a deep dive with us today on endophytes. So Dr. White, I'll let you do any more introduction that you want to do, and then get started.
Thank you, Daniel. I really appreciate it. I appreciate being here. This is an excellent opportunity to talk about endophytes, and I'm so glad you guys are interested in hearing about it. So I'm going to try to share screen now, and hopefully it will work. Okay. I have to...
Yep, there we go.
Okay, you see it?
Yep. Perfect.
Oh, how wonderful. It worked. Okay. So what I'm going to talk about is, in this presentation that I titled The Secret World of Endophytes.
Oops. Sorry about that, Dr. White. I think something happened. We lost your screen share here.
Really? Okay, let me go back. I still hear you, so let's see, I'll try it again. Screen share on, allow.
There we go.
You see it again?
Yep.
Okay, well, I'm gonna go to slideshow again. Do you still have it?
Great. We see it. Sorry, we actually lost your video. I think you just have to turn your video back on.
No, my video's on. So, in other words, you don't see me. You don't see me talking.
Yeah, we don't see you. We can see the slides. So that's.
Can you hear me?
Yes, sir.
You can hear me.
Yes, sir.
Okay. Can you see me?
We cannot, but that's okay. Like if we could just see, there we go. Now we can.
All right. I'm going to go share again. Okay. Is that share working?
Yes, sir. We see it.
Okay. So
I think you just got to click share one more time. We saw it.
So it's not working again. It's bouncing off.
Yeah, it was working, and then we lost it. So maybe just try it.
All right. Hold on. So I'm turned it off. Now I'm going to turn it back on.
Yep, now we see it.
And we'll see if it works. Is
There
that
it
good?
Yes,
Do you see me?
we do.
Okay. Okay, good. So basically, plants use endophytes, and they use endophytes for their own benefit, for various things that I'll talk about, and in various ways, and various kinds of endophytes, and I'll describe that. And one specific type of endophyte or use of endophytes is in the rhizophagy cycle, and this is a newly discovered process where plants use soil microbes to acquire nutrients in the soil. And I'll explain how that works. And then part 3, I'll talk about some other nutritional symbiosis in plants. And if we get there, part 4, I'll talk about the evolutionary legacy of endophytism and basically the deep evolution and of organisms on the planet and what the role endophytes had. We'll see if we get to that part, though. Okay, so part 1, what are endophytes? So endophytes can be fungi or they can be bacteria, be really any microbe that goes into plant tissues. The important thing is that they don't cause disease in plants. And this actually is from a grass. There's a fungal endophytes that go into grasses, and you can see actually in the background the white cells back there. Those are the cells of the grass. And then you see this blue stained hypha. This is the mycelium or the hypha of the fungal endophyte inside the plant. These are in grasses like fescue grass that typically have this certain type of endophyte, and I'll describe that. Endo meaning internal, and phyte meaning plants. So these are microbes that go in plants and don't cause disease, and that's the important aspect of them. They don't cause disease. Instead, they have some beneficial effects on plants, and I'll describe all those beneficial effects during the course of this presentation. So what are some of the effects of microbial endophytes? Just to list them, they'll improve the stress tolerance in plants. So they will make plants so they're hardier and more resistant to things like soil salt or heat or heavy metals. Anything that converts to an oxidative stress, they will help with. They'll reduce susceptibility to oxidative stresses. They will also suppress pathogenic fungi. Okay, so fungi that normally cause disease, when they're impacted by these endophytic bacteria in particular, they'll be avirulent. They'll not cause disease. They'll have less capacity to cause disease, and I'll explain how that works. They modulate root development, and that's just a fancy word to say they control root development. Basically, plants don't grow right if you take away all their endophytes. They actually need endophytes. Plants evolved with these microbes in their tissues, and so they use these microbes in order to develop properly. Endophytes will improve nutrient absorption into plants. Some endophytes actually function directly to absorb nutrients from the soils, and those are the endophytes in the rhizophagy cycle, and I'll explain how that works. Endophytes will alter chemical constituents in plants. They will, for example, in carrots, I'll describe an experiment. With a certain endophytes there, you get more carotenes. Without it, you get less. So the endophyte in the tissues will alter the quality of the plant and the quality of the crop. They will also defend plants from herbivores. Some endophytes will produce chemicals that will cause insects or even animal herbivores to stay away from the plant. They won't like the chemical that the endophyte is producing in the plant. These tend to be fungal endophytes. Okay, and finally, some endophytes will actually inhibit competitor weeds of the plant, and I'll talk about that when the time comes. All plants have endophytes. There are no plants that don't have endophytes. In fact, all plants will have communities of endophytes inside them, and you don't know they're there. you really have to know how to look at them. You have to have a microscope for one thing, and you have to have particular stains to stain them. Otherwise, they're invisible in the plant. So you really have to have the capacity, the tools to look at them. But if you do, you can see them in there. And first, I'm going to talk about these bacterial endophytes. And these bacterial endophytes are actually more common than fungal endophytes. But both are common in plants. But the bacteria tend to be more critical as endophytes in plants. Okay, this is an example. This is a cactus on the desert island of Bonaire. Some of my work, I get to travel, and this is a good thing about my work. I can travel to strange places or exotic places and look at plants there, study the plants there. And so this is one place. This is on the desert island of Bonaire in the Dutch Antilles. And the whole island is covered with this desert in the middle. And they have all these huge cacti called caduceus cacti, the caduceus cactus. And if you look way up at the top of the plant, these cacti produce fruits. And you can see here these purple fruits, kind of like the opuntia cactus fruits. If you cut that open and pull out the seeds, you see seeds like this. This is actually the seeds from one of those cacti. And then these seeds can be germinated in azenic conditions, that is without contaminants. And you get a seedling like this. And this has actually been stained for hydrogen peroxide, one of these stains that we use to look at these endophytic bacteria inside root cells. And here you can see the root here, and you see a shoot above, and you see all the hairs coming out. And you see it's orange. That's because of that stain called DAB. And this is the stain we use, diaminobenzidine, but just DAB. But you see it stains brown there. And that brown actually around the root is due to hydrogen peroxide. And as it turns out, the hydrogen peroxide is the result of an interaction that happens between the bacteria and the plant. And it's basically a chemical interaction that happens. And I'm showing that over here to the left. And you can see the H2O2, that's the hydrogen peroxide. That's the result of the interaction. And so the DAB will actually turn wherever there's hydrogen peroxide brown, so we can see where those bacteria are inside the plants. Otherwise, you wouldn't be able to see the bacteria. Okay, this is that caduceus cactus root hairs, and you can see all these dots inside these hairs, and those hairs are filled with bacteria, and all of those dots are the bacteria, and they're brownish due to the hydrogen peroxide that's around them. This is also counterstained with a blue called aniline blue, with a stain called aniline blue, which also stains these bacteria, and we can see them in there. This is a close-up of one of those hairs, and you can see the pairs. These arrows are indicating pairs of bacteria in the root hair, and these root hairs become full of bacteria. Okay, what about crop plants? So all crops also harbor endophytes. The fact is that many crops have lost many endophytes, but they still harbor some endophytes. If you look, for example, at hemp, and one of my students, April Michi, is working on hemp endophytes. You take some seeds of hemp and germinate that, again under azenic conditions. You get a seedling that comes out. Then you stain that with this DAB, and this is the root hair here over to the left, and you can see all these little brown dots in there. Those are all the bacterial endophytes that are inside that root hair. To the right, you can see the root hair, the kind of the plasma membrane in that root hair, the root hair cell itself inside, and then on the outside you can see the root hair cell wall out there. And you see it's kind of pulled in a little bit from the cell wall, so you see it's kind of constricted there in the middle. But all those dots in there, all those spherical structures, those are all the bacteria that are inside that root hair. So hemp, cannabis, carries these endophytes too. This is a close-up of one of those root hairs, and you can see the bacteria in the root hair. Where do these bacteria come from? They actually come from the seed. So cannabis and many seeds, most seeds will carry some endophytes on the seed surface, and they'll carry other endophytes in the interior of the seed in the embryo or very near the embryo. And I will show you one of those. Okay, and here is Actually, mint is an example. White sage. These are a couple of my students that are working on this mint. And if you look at the mint seeds, the bacteria are actually, in this case, carried both on the surface but also in the seed itself, very near the embryo. And this is the way those mint seeds look like. So some of the microbes are on the surface of that, but basically the seed came from a structure like this. This is an idealized drawing of an ovule of a plant that forms into a seed, and you can see these various tissues. You have out here these two layers that'll become part of the seed coat. And then you have inside the seed, you have in the very center, you have the embryo itself, or the egg sac, it's called. And that egg sac will then develop into the embryo. And you have this tissue out here, this tissue that composed of many cells, that's called the nucellus. And it's very near the embryo. This nucellus is actually where some of the bacteria, some of the endophytes are vectored inside that seed. We'll look at that. Okay, so this picture shows parts of the seed, and you can see to the left, these two layers are part of the seed coat. And then you see the nucellus tissue here in this, all the way to the right, this picture, you can say cellular nucellus. And then you see a little bit further to the right of that, you can see the embryo. And then you see this little line here, it says embryo sac. So this nucellus actually has some bacteria in it, embedded in it, inside that seed. This actually shows one of those nucellus cells. And you can see these little pits where those bacteria are formed. You can see the arrows are indicating those bacteria in those little pits. And this is actually a close-up that shows some of the bacteria, and you can tell they're in rods in those little areas. So these bacteria are already inside the seed. When this seed germinates, these bacteria will infect that embryo. They will go into the embryo, so that they also will have endophytes in them. This just shows some more of that nucellus tissue, and you can see the blue areas where the bacterial endophytes are located. Tomato, another crop. And you think of tomato as a possibly sterile plant, but in fact, it really, at least in the interior, it really is not sterile at all. It has many endophytes in it. And if you take a cut into it, you can see the seed. Those endophytes, actually, those bacteria affiliate with the seed especially. And you can see some tissues associated with the seed where those bacteria will actually associate with. They'll be on that, but they'll also be inside the tomato seed itself, near the embryo. Okay, this is one of the endophytes. This is one called Micrococcus luteus, and you can see the cells form these little tetrads, or like little clusters of 4 cells each. So this is the normal way this cell works, this bacterium works. It has its individual cells in tetrads or clusters. And when a tomato seed germinates, that bacterium will actually colonize the root. And this shows a root of a tomato seedling, and you can see the root hairs over here to the left, and you see the root tip over here. You see right at the very tip to the right. That's called actually the root cap itself, and then you see the meristem underneath that in here, the middle, the area that's not stained. And so the areas where the bacteria colonize are in this area out here, the microbial entry zone. This is stained actually for ethylene, and it shows the area where the bacteria are entering into the plant tissues. The bacteria appear to be producing ethylene, and so we can stain them. We can see that part of the root where the staining is. Now, they also are entering at the root cap, right around here, and you can see this purple area. That's where the bacteria are going in as well, right in this zone. So they're entering 2 parts of that root, actually, of the tomato root. Okay, this actually shows the area of the entry zone. This is when we took that micrococcus from tomato, we put it in a different plant, a different plant, Rumex crispus, which is a very common weed. But you can see right around the area where they enter, we look at the cells, and you can see those tetrads actually inside the cells. So they've already, this is the epidermal cell of the root. You can see they're in the epidermal cells. If you go deeper, you don't seem to see many in here. Every now and then you can see one, but mostly they go right in this epidermal layer on the outer surface of that root. Okay, this is a root, the same tissue that we have over here with the bacteria in it, but we didn't put the bacteria in this plant. And you can see those cells don't have bacteria in them. Okay, this is a close-up of that micrococcus in the tomato root cells, and you can see the tetrads are there, and you can see they're inside those cells. What happens when these bacteria go in, they will, actually the plant will secrete superoxide on them, and I'll talk more about this. This is actually part of the rhizophagy cycle. But the important thing is that will cause these bacteria to change their shapes. And they actually will lose their cell walls, and they'll form these little spherical protoplasts. And so that happens after they go in. So they'll go from the tetrads to these little spherical protoplast phases. And these arrows here indicate that. And you can see these little spherical things stained various colors, but they're actually inside those root cells. Here you see them also. They're in the place actually called the periplasmic space. Is just underneath the cell wall, but outside the membrane, the plasmid membrane of the cell. And you can kind of see that here with these protoplasts. You can see the membrane kind of variable up here at the top, and you see those protoplasts, those bacteria actually on those. So their shape changes once they go inside these root cells. This shows a tomato, I think it's tomato, root hairs. And you can see outside, you can see the micrococcus, you can see the tetrads out there. But inside, you can see the spherical protoplast phases that are staining inside those root hairs. This just shows a root hair developing, and you can see those protoplasts in there as they develop. This shows these microbes in the root, actually the root hair, and they will actually come out of the root hair. And you can see here to the right, you actually see the tip of a root hair, and you can see these little white arrow indicating these spherical structures. Those are the protoplasts, and you can see little channels through the root hair. And you can see on the outside, once they go out, they start reforming their tetrads. So inside they're naked protoplasts, then outside they form their tetrads again. Here you can see to the left, you can see it's a root hair, and you can see the little bacteria coming out of the tip. You can see the channels there, and you can see as they mature, they fall off and they reform their cell walls there. So they have these two forms. This happens to be that rumix seedling again. You can see it, that common weed. Now, you can lose endophytes from the seed surface. In fact, we think that most cultivated plants have lost endophytes. But you can make the matter worse if you treat plants or treat seeds in a way to kill the microbes on the seeds or in the seeds. And so an example here is cotton seeds. So you have on cotton normally grows with these fibers all over it. And it turns out that microbes are vectored on those cotton fibers. And what we do, once the cotton is removed, you get these seeds that have little bits of cotton fibers on them, and then those are dunked in acid. And so that all the the cotton fibers are ate away. But that eating them away removes also all the microbes. You can see here without any fibers, but there's also microbes. They're very poor in their endophytic microbes that they're vectoring on the surface. And what that means, and here actually I could show you here. Here's some of the fibers, and you can see it's microscope. You can see this little granular structure in here. Those are bacteria on those fibers. So eating those away removes those bacteria. Here you can see it again on the fibers. And here you can see on the surface of the seed coat, actually close on the pits on the surface, you can see that granular pattern again. That's where the bacteria are on that seed surface. And removing those microbes really means that the seedlings that develop are weak and not very hardy. They are susceptible to diseases. They don't have the microbes that they need to go out and get nutrients. And they really require fertilizers, and they require insecticides and pesticides. They're not very hardy without those microbes. And that's compounded because much cotton is actually grown in soils that are poorly managed in terms of biological diversity. And so that means the cotton plants cannot really get back their endophytes from the soil. They can't fish them out of the soil to kind of repair what we've done with this acid diluting. Other seed treatments also hurt plants. Oftentimes in plants, husks like Bermuda grass. Normally, these plants have a husk on them. It turns out the bacteria in the husk. So when you remove the husk, that makes the seed more vulnerable because it loses its endophytes, its beneficial endophytes. And so it's necessary then actually to put fungicides on the Bermuda grass seeds that have been dehusked in order to protect them from pathogens because those protective endophytes are missing. So it's dangerous whenever coatings are put on seeds. Unless you're putting microbial coatings, you may be harming the seedlings. Okay, this is another system that we looked at. This is English ivy, and just kind of to illustrate the point of these endophytes. With English ivy, there is a bacillus that's vectored, and the bacillus that actually goes with the seed and goes with the fruit. Normally, when the plant germinates or the plant grows, the seed germinates and the plant grows. The plant will grow normally and will be resistant to disease. And this is an experiment where all the microbes were removed from seeds, and then they were put back on some seeds, and they were left off of other seeds. And what you can see is where they were put back on, you see this English ivy plant grew very healthy, no disease there. But where they were left off, you can see there's a disease here. And that disease happens to be an alternaria, which causes this leaf spot. When the bacillus is present, the plant is protected, defended from these pathogens. So it's a defensive association here with this. Also, the plant seems to be growing better over here to the left. Okay, and what happens is this bacillus normally will go all over the plant and in the plant, but this actually shows below here shows English ivy leaf surface here, epidermal cells below, and you can see these little rods or these little things in here. Those are the bacteria all over the surface of those leaves that are developing. So the plant becomes covered with these bacteria, and it forms a kind of a defensive shield for this plant. And this is, in this case, the bacterium becomes a kind of bodyguard. These endophytes become a bodyguard for the plant. And you can see here the bacteria in the middle, and you can see the fungus on the periphery, and you can see there's an inhibitory zone. This has been called the bodyguard hypothesis by some people, that these microbes protect plants from diseases and pathogens and so forth. Keith Clay, who's now at Tulane University, coined or developed the idea of endophytes as defensive mutualism. That is, they pair with plants to defend them from various insects and animal herbivores and possibly diseases. The way that these endophytes protect plants is they will, in order to inhibit these microbes, fungi, because they produce special compounds, antifungal or anti-insect compounds. In this case, Bacillus will produce these lipopeptides, and you can see one of them here. And what these lipopeptides do is they will go into fungal hyphae and they'll cause it to leak nutrients. And that will cause these fungi to lose energy because the bacteria will essentially take it from them. And that makes them avirulent. It makes it so they can't cause disease in the plants. Okay, another way that they will protect plants is they'll produce, for example, hydrogen cyanide. And this hydrogen cyanide will inhibit fungi. It inhibits their respiration and will cause fungi to leak nutrients again, actually. It'll weaken them. Okay, and some endophytes actually will have herbicidal effects for plants, and we have termed that phenomenon endobiome interference. And that is, the endophyte, the bacterial endophyte, will go out of the plant, its host plant that it promotes the growth of. It'll grow into the soil, and then in the soil, it'll colonize other seedlings, and then it'll go into their tissues, and then it will inhibit the growth of those other plants, and it'll go into the endobiome of the other plants and inhibit their development. And an example here is shown here, and this is actually shows dandelion. And what we did was we took an endophytic bacterium from common reed grass, this invasive Phragmites australis, and then we took it and then we put it into dandelion. Here's how dandelion grew without the big normal growth, without the bacterium. Then we put it in the plant, and you can see the dandelion seedlings were highly repressed. So it interfered with the growth and development of. So you can imagine if there's huge stands of Phragmites growing with this bacterium going out into the soil, it's going to inhibit the growth of dandelion and probably a lot of other plants that come into the area. So where do these endophytic microbes come from? I want to emphasize they come from soil. Initial place is always soil. Most of these microbes actually are soil microbes that are taken into the plant by the plant. The plant picks them up for its own benefit. The plant essentially fishes those microbes out of the soil. But then the plant will put some of those microbes that are beneficial on the seeds so that the seedlings can then be protected by those microbes and stimulate growth of the seedling and be sure that the seedling has a good start. And what about fungal endophytes? Fungal endophytes, mostly I talked about bacterial endophytes, but fungal endophytes are also there. And fungal endophytes can be yeast types, and they can also be filamentous fungal types, and I'll talk about both. This is a yeast type. This is one called a black yeast. This is Areobasidium pullulans, a black yeast. You see the yeast cells here to the left. This is common in some plants, very common as an endophyte. People who are studying endophytes isolate these things fairly frequently. These also will go into root cells. And when they do, they will change their shape. They will form these naked protoplasts again. Only with fungi, they have a special term called mycosomes, and they will lose their cell walls and they'll form. This is actually from an article done by Peter Atzat and Michael Whiteside at Davis, California, but where they showed actually that these fungi, these black yeast, will go into cells and then they change their shapes. Okay, this shows some of those in root cells, and this is a plant called snake root. But you can see the root cells here. This top cell actually shows these mycosomes or fungi inside, fungal protoplasts inside that cell. And you can see they're also with bacteria because the one below it actually shows bacteria inside the cell. So you can have both fungal endophytes and bacterial endophytes in the same plant. And that's an important principle. Now, are there any bad endophytes? Okay, there are no bad endophytes for plants. They're all good for plants. But are there bad endophytes for animals that graze plants? And there are in forage plants and plants that animals tend to eat. We don't have these kind of bad endophytes for any food that people consume, but for animals, grazers, for example, there are some really bad ass endophytes, if I can say. These endophytes really are producers of toxins. And some of these endophytes, some of these fungal endophytes are present in cool season grasses like fescues and ryegrasses and some other cool season grasses, and I'll talk about those. They're also present in loco weeds. Loco weeds, they're these legumes that tend to grow out west. And for a long time, it was thought these loco weeds produce toxins that make animals crazy, that'll make them go crazy, it'll poison them. But as it turns out, it's not the local weed at all that produces the toxin. Instead, it's an endophyte in those local weeds. Morning glories also will have endophytes in them that will produce toxins, and it's the same situation. Some morning glories are poison, some morning glories are not. The ones that are poison all have endophytes in them. So this is a really fascinating area of study. And for grasses, they're actually called epichloi endophytes, and I'll talk about these. But these epichloi endophytes produce mycelium that goes into the leaves of the plant. They go into the seeds of the plant and the shoot and the stems of the plant, of the grass plant. But they don't go into the roots. At least they tend not to go into the roots. So this shows one of these epichloi endophytes in actually tall fescue, and you can see in the background those are actually the cells of the plant. This could be in a leaf or a stem, for example, and you can see the hypha, this convoluted, this kind of wavy structure. That's the hypha, the mycelium that occurs in between the cells of the plant. So it doesn't actually go in the cells of the plant in this kind of endophyte. Instead, it's just between the cells. But what they do is they will fill the plant with alkaloids, these ergot alkaloids, and make the plant toxic to animals. Okay, and just, for your edification, the spelling of epichloi is this, epichloi endophyte, and that's actually the scientific name, the genus name. And these are in the family Clavicipitaceae. And this is actually an ergot, the family of ergot, the ergot fungus family. And these are Ascomycetes. And these endophytes will produce alkaloids. They produce all of these kinds of alkaloids. Ergovaline here at the bottom. Okay, this ergovaline, this is actually an ergot alkaloid. These are highly toxic to animals. Lolotrims, these are also toxic to animals. These will cause like a staggers, twitching, constant twitching in the animal, a nervous kind of activity. Tends to be less fatal, but will affect animals so they don't eat and they appear very sick. These lolines, informal lolines, these are anti-insect chemicals. Pyramine, anti-insect. So you have all of these kind of chemicals that these endophytes can produce. And those chemicals will, some will deter feeding of insects, and others will cause toxicity in animals. And so you see here some of the diseases or the syndromes in animals, one called fescue foot, that happens on tall fescue in cows grazing tall fescue. Summer syndrome, also in tall fescue. Ryegrass staggers, that's in ryegrass. Animals, all kinds of animals will get that, and it's kind of an involuntary staggers, twitching, kind of nervous, trembling kind of effect in the animal. And then you have something called sleepy grass narcosis. It's really cool. Occurs in wild grasses, occurs in the Americas, in South America, and Central America, and in North America, in the mountainous regions. And then in South America, there's huecu toxicosis, or also called timbladera. And I'll talk more about some of those. They're really cool. That's because these endophytes are producing these chemicals. And this is actually the fescue foot, and it's due to the chemical called ergovaline. You see that listed at the bottom. But what happens with this ergovaline and all of these ergot alkaloids is they will cause vasoconstriction in the veins. And that means the veins will be constricted, and they'll cut off the flow of blood to the body's extremities and make the reduced blood flow through those constricted veins. And so then the parts of the body, like the hooves and the tail, no blood will go there, and so they'll die essentially on the animal. And you can see these hooves that appear to have these lesions on them, and those hooves or tails will just rot off of these animals. This is the same chemical that is in ergot, and ergot affects animals, but it also affects humans. Ergot is not actually an endophyte, but it does infect grasses, and it's in the same family, produces the same chemical. And ergot historically has caused toxicity in humans, And it basically, this is the ergot. What it does, it will form, the fungus will infect these seeds of a plant, typically rye or wheat or even fescue grasses out here, wherever you have kind of cool season grasses. Ergot will infect it, and you'll see these black sclerotia that are formed. These are sometimes simply called ergots, but these black sclerotia are filled with ergot alkaloids. And those alkaloids are all kinds of ergot alkaloids, and I'll show you there's a great diversity of them. But they would get, essentially, this is rye, and what happens is they would get mixed up in the grains. You can see them here in the middle, and you can see the grains here with them in it. And people would eat the grains plus the ergot and grind it all up, and so they would get this bread that was full of these ergot alkaloids, and that was essentially toxic bread. And then they'd eat that, and they'd get the same kind of effect that those animals get. These are some of the ergot alkaloids that are present in the ergot, and you can see some of the names. Ergonovine, we use that as a medicine. But you also see D-lysergic acid, sounds very close to lysergic acid diethylamide, LSD. And that's because these were actually used. This is actually what Albert Hofmann used to create LSD. And I'll just mention that just briefly. But here you see in humans this ergotism. You see the same thing that happens in animals because of that vasoconstrictive effect. We don't have any endophytes in our crops that do this, so you don't really have to worry about this in terms of any endophytes in any of the food crops that we're producing. But as I mentioned, Albert Hofmann took these ergot alkaloids and he varied them and produced our LSD-25. He actually used ergotamine, that one that I mentioned in that list. And this just shows LSD here at the top, and you can see from ergotamine in Claviceps purpurea. Claviceps purpurea is the ergot fungus. So this is fungus, and this whole family of fungi is pretty famous. And Albert Hofmann, there's a poster for him. This is shows, if people were very grateful for everything he did for the psychedelic era. And there you see a blotting paper, LSD blotting paper, Albert Hofmann, signed by Albert Hofmann. Look at that number, 22 of 60. Well, these endophytes in grasses, different kinds of grasses, have different kinds of endophytes. And here's one in ryegrass that actually will produce lolotrim B, and it doesn't produce ergot alkaloids. And this lolotrim B is this big compound, big chemical, but it makes these animals, horses and anything that eats it, it makes them have this staggers, kind of involuntary tremor effect. And I've been told that for horses, it ruins horses, that once it happens to them, they never will want to be ridden, or they are always affected by that. I guess it's like, it's like a bad trip if they get affected by ryegrass staggers. There's another one in South America I mentioned called huecu grass, but it also is called tembladera down there, and that's from the Spanish word that means trembler. And huecu actually is an Indian word, an indigenous word in South America that means intoxicator. And basically this grass grows in large areas, huge expanses of areas. And if animals eat it, they will sicken and become as if they were intoxicated, sick, and they'll be sick for days. And so they're developed down there in South America something called the strategy of huecu by the Indians. And so when the cowboys or the soldiers were down there, the same thing happened in South America that happened up here in North America. The indigenous peoples were pursued by soldiers, and they would go into the areas with their horses to escape the pursuers. And because the indigenous people knew about these grasses, they wouldn't let their horses feed. So they would go right through that area without letting their animals feed. But the pursuers often didn't know about the huecu grass or the tembladera grass. They would let their animals feed, and then that was the end of the pursuit right there. That ended it. The strategy of huecu ended the chase, and they could get away. This also happened. Bandits also did the same thing. In North America, in parts of Mexico, in Central America, you have another grass that we call sleepy grass, actually, in North America. And it occurs in the mountains in North America. And one area is around Cloudcroft, near Cloudcroft in New Mexico. There is a place there called Sleepy Grass Campground. You can see the sign here, Sleepy Grass Campground. It's called Sleepy Grass Campground because there was a grass there that people discovered was a sleepy grass. It would cause horses to go to sleep. And so what people traveling through those mountains would do is they would stop and rest and let their horses eat the grass. And they would sleep for days and then gradually wake up, hence Sleepy Grass Campground. Itself doesn't produce any sleepy chemical. Instead, it's an epiploid endophyte in the grass that produces lysergic acid amide. And lysergic acid amide has the effect of causing sleeping in animals. It's said that in Central America, some of the indigenous people would use seeds of sleepy grass to feed their babies to get them to sleep. So I don't know. This is all old literature where that was indicated, but apparently it has been known for a while until and thought it was just a grass. But about 40 years ago, I discovered that in fact there was an endophyte in that grass. There's one in South America, in the Transvaal region, actually, that the people called dronk grass, and the Dutch word dronk grass. It's a Melica decumbens, a Melica species, but it contains another epiploid endophyte that produces alkaloids that cause these animals to have that drunk-like, intoxicated-like effect. And every continent basically has it, I guess, except Antarctica. Maybe Antarctica has some, I don't know. But there's one in China that actually they call drunken horse grass. And again, they knew the grass was toxic before they knew there was an endophyte in there. And I have Chinese collaborators that are working on this grass, and they've published, I can't tell you how many papers on drunken horse grass and some other endophytes that they have. So it's a big area now. But these fungal endophytes, okay, besides from that negative stuff, they're actually positive things. And the positive things have to do with disease protection. And this actually, so for turf grasses, these endophytes, the kind of endophytes are actually selected for in turf grasses. And so this is a plot, this is something we discovered at Rutgers-University, a plot of a fine fescue grass. And the one to the left has one of these endophytes in it. And the one to the right lacks the endophyte. And you can see all this brown in there. All that brown is due to a disease called dollar spot disease. And it turns out the endophyte protects this turf grass from this kind of disease and several other diseases. People have also tried to find endophytes that are non-toxic and put them in forage grasses. And they have actually, a group in New Zealand has actually been successful at that. And what they did is they tried to find endophytes that produce the anti-insect chemicals that I described early on, and that don't produce the toxic animal, the alkaloids, the ones that affect animals. So, and they found those, and then they put those in tall fescue, and they put them in ryegrass, and I think this one is for tall fescue, I think. And they named their endophyte, in this case, their non-toxic endophyte, Max Q. And there's a toxic endophyte that they refer to here as Kentucky 31 wild type endophyte. And you can see animals grazed on Max Q get bigger because they're non-toxic, no toxicity. And on Kentucky 31, they're smaller. And then you have also the benefits of anti-insect, and also there's other positive effects. When you have these fungal endophytes in grasses, they grow better, they're more stress tolerant, and more resistant to fungal pathogens and so forth. So that's the benefit of, and so people have developed products for that. This is one of the investigators on fungal endophytes. His name Rusty Rodriguez, and what Rusty did actually is to study some fungal endophytes and some wild grasses and take those and put those in cultivated plants. And what he found is that endophytes are adapting plants to specific habitats, specific stressful habitats. Like, for example, one study he did has to do with dune plants and salinity or salt, the stress imparted by salt. And what he found is that in plants that grow in the salt, they would associate with certain fungal endophyte, a fusarium species. Up here you can see the list. That's the name of the fungus there. You can see the word spelled out here, fungal endophyte there. And then that gave the plant tolerance to the salt stress. And so you can see an experiment here. To the left was without the fungus, and you can see in these little containers, you see increasing levels of salt, and you see the plants doing worse. But here, when he had his fungi there, you can see the plants could tolerate more of that salt. He called that habitat-adapted symbiosis. He went on to make a company, he and his wife, Regina Redmond, I'll show another experiment they did. They went on to make a company where they're marketing these kind of endophyte products. Regina Redmond, she's here, is another investigator on it, one of their studies, Luis Marquez. But they did a study at Yellowstone National Park at these thermic soils. These soils that get up to 70 degrees Celsius centigrade, get really hot. And what they found there is that this plant happens to be a panicoid grass, a warm season grass. If it had an endophyte curvularia here, curvularia protuberata, you can see it could withstand these high soils, these high temperature soils. And you can see to the left here, plants with the endophyte in those thermic soils. You can see they're all green, mostly green. And then without the endophyte, you can see to the right, they died. Later on, they actually found that there's also a virus involved in this interaction. The virus has something to do, apparently, with that resistance to the stress. Okay, so just in conclusion, all plants have endophytes. Those endophytes are universally beneficial to plants. There are no negative endophytes. There may be some microbes that go into plants that have no effect on plants, but they don't tend to be pathogenic. Those microbes tend to be selected against for naturally in plants. And they can be fungi and they can be bacteria. They can also be green algae, for example, or algae, other kinds of algae. Sometimes you see algae going into plants, like, for example, rice in wetland, in wet habitats. You'll sometimes have algae in there, green algae that fix nitrogen, actually. And that plants get these endophytes from soil. Most of these come from soil. The negative kinds that I talked about, the epichloi endophytes, they don't come from soil. But most of the endophytes do come from the soil. And that some of these, like epichloi endophytes, are bad for animals that consume the grazing plants, and also like loco weed. Those have endophytes that are bad for animals, but otherwise they're really good. Okay, so questions? Anybody have any questions at this point?
All right. Can you hear me?
Yep, barely.
Perfect. All right, I'll try to stay close to my microphone here. Great, thank you, Dr. White. That was fascinating. We've had a lot of chatter going in the chat here. So also, before I get into some of the questions here, just want to let everybody know we're actually this time around, in case you were at the last event, this time around we're just going to have 2-hour sessions total. So give you guys a little bit more of a break in between. And Dr. White, I know that's a little bit of news for you, so we'll just end up shortening this presentation a bit. But we will get into Q&A now. Are you still hearing me okay?
Yep.
Okay, perfect. So first question, can you stain any endophyte to see if your roots are colonized?
Yes, you can. We have different kinds of stains. Aniline blue is one good one that we use. Also, these reactive oxygen stains, they're pretty easy to use. And that's just you could buy these tablets from like Sigma or Aldrich that contain diaminobenzidine, and you can stain those overnight. You can see bacteria and and fungi inside the plant tissues. But aniline blue works very well for fungi.
Awesome. Let's see. Somebody asked, what about PGP producing endophytes? How do they influence the plant and its systems?
Yeah, the plant growth promoting endophytes, most of those are bacteria. And they actually do go into the cells in the rhizophagy cycle, which is what I'm going to talk about next. And they will get nutrients for plants, and they will make plants more hardier. They will promote the growth of the plants. Generally people think of plant growth promoting microbes as producing hormones, and that's the reason that they work. But in fact, they actually carry nutrients as well.
All right. So somebody asked, if you use big ag fungicides, how does that impact endophytes?
Yeah, big ag fungicides in general are going to impact certainly your fungal populations of endophytes, but they also might have effects on bacteria. I mean, you have to check, but they may be negatively impacting your microbial populations in general, your microbial communities on plants in general. But sometimes I understand you may have to use some kind of a fungicide if you've got some disease going. It's just that I think you should be probably conservative if you use it. Try not to use, certainly not to go to initially what you want to do. You don't want to routinely start using fungicides all over your plants.
Mm-hmm. All right. If we treat cover crop seed with inoculant, then would they produce seed with endophytes?
If you have the right microbes, then you should get if they're beneficial to the plant. If they will colonize the shoot tissue, it really depends on the microbe, but the answer is yes. In some cases, if you treat plants with microbes, you should get them onto the seeds of the plant. I've known a case. I have a friend who works with organic breeding of corn, and he used some growth promotional microbes on his corn a few years ago. And last year, we actually examined his seeds and seedlings, and we picked up the same microbes coming off of those seeds. So they were vectored, after a number of years from his initial treatment to the kernels, the corn kernels. So they were staying with the plant. They ended up into the seeds. Yeah.
Awesome. Yeah. The last part of that question, they were asking if you have to continue inoculating. So it sounds like you don't necessarily. It sounds like they will stay with the seeds most likely.
So they would continue to benefit the plants. But you have to check and make sure that that's the case, right? Just any microbe some microbes may not stick with the plant. You can get some growth promotional microbes and put on the plants, and they'll go onto the roots and they'll kickstart what this process I'm going to talk about called the rhizophagy cycle, but they don't necessarily stick with the plant long term. So you have to really check.
Got it. All right. So somebody asked, is there any research relating endophyte colonization to the human gut microbiome? I think there is, right?
That is the coolest question.
Mm-hmm.
Yeah, there really is not, but it is correct that humans are picking up gut microbes from the foods that we eat, from the plants that we eat. And it's known that humans who have a broad diversity, like people who live in the rainforest, have a broad diversity of plant foods, have also the most diverse microbiomes. And that's because they are consuming all these plants with all their endophytes in those plants. That's neat. Some of these microbes are the same ones. Some bacillus species on plants go into the gut and are also found in the gut. So, and other bacteria. I mean, many of these bacteria are the same ones that we have in our guts, and certainly many animals have in their guts.
Mm-hmm.
Yeah, it's a good question.
Yeah. Yeah, awesome. It's fascinating. So somebody said, I'm curious how long we've been doing that process to cotton seed. So you talked a while back about doing something to the cotton seeds.
Repeat that again, would you please, Daniel?
Yeah, sorry, and maybe we might skip this because I'm not sure actually what it was referring to. But I think you had talked about some sort of process being done to cotton seed, and so
it's called acid delinting. Acid delinting, and what happens is they soak cotton seeds in hydrochloric acid, which eats away everything on the surface, and they do that so it goes through the planter, I should say. We do that just so we could plant the seeds. And so all of our seeds are delinted like that. It's better if you're going to have organic cotton to not do that.
This person was asking how long has that kind of been like a standard procedure?
How long does that affect?
I mean, like when did that start coming into practice? Was it like 10 years ago or 20 years ago?
Gin.
Really?
That's been since the cotton gin was invented. Yeah.
Wow.
We have done that a long time. That's why cotton is such a dirty process. Dirty crop, because you have to have so many chemicals, fertilizers, and everything else to grow cotton. It's because we've devastated the microbiome with our cultivation practices.
Yeah, makes sense.
Yeah.
Wow.
Daniel,
all right.
Shall we go on now?
Yeah, we did have a lot of questions. Let's get a few more in here and then, we'll keep going. Let's see. What is the distribution of helpful endophytes between, aerobic, anaerobic, facultative anaerobes? Is there?
Nobody knows that answer. I mean, there could be some endophytes that are, and we don't even know if there are, for example, archaea in there. I mean, some people have suggested we have archaea, but we haven't isolated any archaea. They don't isolate. Mm-hmm. Some microbes may not be isolating. We're only seeing what we isolate out. So we can't really answer that question.
Okay. And then, are there any known viral endophytes?
Known virus endophytes?
Yeah.
There is the study that, I mean, the answer is no, but in a way, yes, because the study that Regina Redmond did on that thermic soil at Yellowstone, they later found that there was a virus involved. They called it a tripartite association. So it was the fungus, it was a virus in the fungus. And so long as the virus was there, they were seeing this protective effect of the plant. When the virus was gone, they lost it. But it was a fungus virus. Mm-hmm. But you don't see that frequently.
Yeah. Okay. And then somebody's wondering, and I think this is especially relevant to some people who use Korean natural farming techniques and are making fermented plant juice from their plants. Someone was asking if grass has alkaloid endophytes, then is using that grass bad for our plants if you were like making a fermented plant juice?
No, the answer is no, it would not hurt. It would not. These chemicals don't hurt plants at all. They tend to affect animals that eat the plants. But when those alkaloids go into the soil, they actually become they're made of nitrogen, part nitrogen, and they become nitrogen for microbes. So no, I would not worry at all about that.
Great. All right. Yeah. Let's keep going in the interest of time, and we've got a clock down in the bottom there, so you can keep an eye on that. Okay.
Here we go. All right. So part 2. What is the rhizophagy cycle? So with the rhizophagy cycle, and what I'm going to try to convince you of here is that plants actually, in the rhizophagy cycle, plants are cultivating microbes, just the way we cultivate plants. That is, plants are farming microbes in the soil in the rhizophagy cycle, and that's the basic tenet that I'm trying to get across here. And this it was observed back in 2010 by some Australian investigators that microbes were growing into root cells. And this is one of these investigators. This is Channy Pongfu. They were in Queensland, Australia. And what they found is that some plants were actually taking in yeasts and taking in bacteria. And what they did is an experiment where they labeled these microbes with the green fluorescent protein so they could see them inside the plant. And here you could see root hairs. You can see all these little green dots. Okay, those actually are yeasts inside the root hair. That's an E, that E below. That's a root hair with all the yeasts in it. You see they're fluorescing. Also, if you look at D, you can see those are root hairs there. And you can see all those bacteria, in that case, bacteria fluorescing inside the plant. And what they found was that in time, in 3 or 4 days, the fluorescence stopped. And what happened is the plant appeared to be degrading those microbes. And then they later, in a chapter after this article, they termed this process rhizophagy or rhizophagy, if you want. Some people pronounce it rhizophagy. But rhizo means root, phagy meaning eating. So root eating, to convey the idea that plants are actually eating these microbes. And what we found is that this is actually, the rhizophagy process is actually a cyclic process. We called it the rhizophagy cycle, but to denote that it's cyclic. And what happens is that plants at the root tips secrete sugars, they secrete exudates, they secrete sugars, they secrete organic acids and sometimes amino acids. And that attracts microbes to the root tip in that exudate zone, and then they start to grow there. And then the plant will take those microbes into the root cells. And then the plant, once they're in there, the plant will hit them with superoxide. Superoxide is highly potent, and it oxidizes away the cell walls of the microbes, and it starts to extract nutrients from the microbes. Microbes that survive that process, that are in the root cells, then trigger root hair elongation. And as those root hairs elongate, microbes will be shot out or ejected back out into the soil. And once they're into the soil, then they'll reform their cell walls and they'll go out and acquire more nutrients and then be attracted back to the root tips. Okay, so that's the rhizophagy cycle. In the soil, the microbes acquire nutrients, soil nutrients, macro and micronutrients, and once they go into the plant gets those nutrients from those microbes and then sends them back out into the soil to get more nutrients. So it's a cyclic, continuous process with certain microbes involved. Okay, this is a grass seed, and you can see the seed is germinating here. You can see the kind of yellow around the seed. The yellow, actually, where these arrows are, is bacteria. And so bacteria will, of 3 or 4, of really a small community of microbes that are vector on the surface of that seed, will then colonize the shoots and the root and go into those cells, especially in the root. But they also will go into the shoot tissues. This is a diagram that actually shows once that root comes out, you can see the meristem here is at the base, and then you have the exudate zone. That exudate zone is where this plant, the seedling, cultivates those microbes. And this rhizophagy cycle, and all of this is happening at the root tips. So this actually shows a grass, a corn roots, and you can see all these little tiny tips. So some plants do this rhizophagy cycle a lot at every little tip that you see. Grasses tend to do it a lot. Some other plants may not do it as much. Taproot plants may not do it as much. This shows an actual grass root, and you can see the microbes out there in a kind of a fog, a little cluster of microbes, a haze of microbes out there. Those are all the bacteria out there stained blue, and they're being internalized at the root. This is right here, the root meristem right here, and so they're being internalized just behind that root meristem. They're going in. What happens when they go in? First, they have their cell walls, and this illustrates that. You have rods with their cell walls. They're hit with superoxide, reactive oxygen, and then they lose their cell walls, and then they form these little spherical cells. So they lose their shape, transformed when they go in. And I mentioned that with certain kinds of endophytes before. And once they form these protoplast phases, they leak nutrients very easily. They leak nutrients, and also they replicate very rapidly inside in chains, whereas if they have their walls, it's slower replication. These replicate very fast inside the root cells, these protoplasts. The plant secretes superoxide from the plasma membrane from enzymes. This is one of those enzymes called NADPH oxidases, NOx, essentially NOx enzymes. These are all in the plant membrane, and that's where the superoxide is produced and put onto those microbes. This shows the root of Phragmites. You can see the root cell here. You see the arrows showing these clusters of bacteria. And you can see, and these are essentially inside these cells. And you can see the small one here at this lower arrow. That was the youngest one, and you can see it stained blue. That blue is because it has protein. You can see these older ones, they're not staining, and that's because those older ones are breaking down because that superoxide, that reactive oxygen, is going in and taking their protein and degrading their protein, and so the plant is getting their protein. This is a diagram of a root cell just to show you where these bacteria actually are going. And this gray out here is actually the cell wall of the root cell. And then this white is actually the cytoplasm of the root. So these bacteria are actually not going in the cytoplasm. Instead, they go underneath the cell wall, but just outside the cytoplasm. And that's where the plant cells secrete superoxide onto them but something else is going on, which I'll show you. They're being circulated in the process of cyclosis, it's called, but that moves these bacteria around and around and will break the bacteria up, but it also encourages extraction of nutrients from the microbes because of that cyclosis. And I'll show you actual films of that. Okay, this shows a tall fescue root hair, and this just shows the blue staining is where superoxide is. And this shows all these little spherical things. Those are all the microbes in here, in those protoplasts. And the blue is superoxide. So the root cell is secreting superoxide onto those microbes to extract nutrients from them. This just shows, this is a fluorescent microscopy. You can see the bacteria, the microbes in those developing root hairs. There you see again, beginning to cluster at the tip. That's important because that's where they cluster at. As it turns out, plants will not grow right without these microbes. They require these microbes to develop properly. And that's probably because plants evolved with these microbes. So they evolved to interact with microbes in their development. And these microbes trigger, one thing that they do is when the microbes are present, plant roots will have this gravitropic response, and that is they will grow down with gravity. Okay, but if you take all the microbes away, the roots will either grow in the air or they'll grow on the surface, but they tend not to grow down. The other effect, or one other major effect, is that without the microbes in the root hairs, within the root cells itself, you have no hair elongation. So you get no root hairs on roots. Those microbes are necessary to make root hairs. I consider again the tomato, and with the tomato, we have endophytes that are already in there, but we've also isolated endophytes from tomato and done experiments with the endophytes. Okay, this actually is an experiment where we removed all the endophytes from tomato seeds using antibiotics, and where the seeds were not treated with antibiotics, and to the left, and you can see the seeds germinated. You can see the roots that came out here, and you see hairs all over there. That's with bacteria. And to the right, you see there's no bacteria there. I mean, there's no root hairs there, and there's no bacteria in there. Those bacteria are critical for development of root hairs. And basically, these are a close-up of these root hairs. And you can see to the right is actually a root hair initial where there's no bacteria. And what we did was we treated it with antibiotics. We also used Clorox to remove all the microbes on those seeds. So these are pretty clean. But you don't see any bacteria inside that root hair initial. Those root hairs without bacteria don't elongate. You have these primordia, you have these initials, but no bacteria there, they get no elongation. Compared to the left, this is with bacteria there, and you can see the bacteria in the hair. You can see it beginning to elongate out. B here, you see them elongating, and finally C when you can also see that the microbes are increasing in those hairs. You can see all the microbes in the tips of those hairs. Okay, so one thing we're really interested in is understanding why root hair growth is linked to presence of these bacteria. And one of my students working on this is Ivy Chang. And this is actually a grass in which all the microbes have been removed, and you can see there's no hairs there. And then she put a microbe back on, and you can see now hairs forming or developing there. And you also can see little spherical dots on those hairs. That's actually bacteria coming out. And our most recent hypothesis from our latest data is that these microbes are secreting ethylene. And that ethylene is actually a plant hormone that makes plants grow. And it, in the case being produced by these microbes, especially the microbes in the tips of these hairs, it makes those hairs elongate. And so they elongate so long as the microbes are there. When the microbes are all gone, they no longer elongate. And this is actually the process, the chemical reaction for that interaction. And that interaction involves ethylene and involves superoxide. The ethylene actually will trigger both cell growth in the plant, but it also triggers the superoxide formation. And then the superoxide and the ethylene combine to form hydrogen peroxide. And that's what we're staining, and that's what we're seeing. And one experiment that suggests this is that the enzyme that produces ethylene is an enzyme called ethylene synthase, and it uses arginine. And so we used an inhibitor of arginine, or an analog, arginine analog called L-name, that is similar to arginine, but it won't work with this enzyme. And so we put that in the system, and what happened with this L-name, or this, it clogged up the enzyme, it stopped working, and we stopped getting ethylene. We stopped getting cell elongation, and we also stopped getting superoxide formation, and we stopped getting hydrogen peroxide formation. So we're leaning towards ethylene as the main stimulant of root growth at this point. This actually shows the tomato root, and you can see here, this is where the bacteria go into the root. You can see the hairs back here, but you see they're entering here, and you can see they're staining. Actually, this is stained for ethylene. You can see staining there, right where they're entering. Another place they enter is right here at the root cap, and you can see they're going in. That's where they go in there, and they're staining there. So this is some of those root cap cells. You can see the bacteria there. You can see the purple. That purple is for the ethylene from that ethylene stain. That's just a close-up of that. That shows bacteria themselves staining blue for ethylene. Again, you can see a primordial. You can see the bacteria in that root hair primordium, and it's blue. That's the ethylene. There's a root hair. You can see the blue again from ethylene staining, close-up of that. So we're still testing that, and we're still going to continue to test that. But I'll move on. This is a Bermuda grass, and this Bermuda grass, you can see this. We've removed all the microbes from this grass and germinated it, and you can see the root here. You see no hairs at all on that root. Here's where we put the microbe back on, put a microbe back on. This happens to be a pseudomonad from a different grass. We put it back on, and you see right away you get hair formation. The microbes are critical for hair formation. When the microbes go in, you can see this is right around the meristem, right where they're entering, and you can see they have the rod shapes there. So they just went in. They haven't lost those rod shapes. This is later on. You can see part of the root, all these brown dots in there. All those are bacteria inside those root hairs. And if you look at the root itself, you can see little brown dots. Those are the bacteria inside those root cells. Here too, this is a root hair. You see of that grass, and you can see all these little bacteria inside it. They're full of those microbes. So these root hairs actually will eject those microbes from those hairs and put them back in the soil. And the way that works is the root hairs work like a machine. They essentially constantly move those microbes around and deposit them out here at the tip of the hair. And the dark line on the outside is actually the cell wall of the root hair, and then you see the dotted line is the membrane of that root hair and the cytoplasm in the center. So these hairs move the microbes around in something called cyclosis, moving them around. And then at some point there is an expansion that starts at the base of the like a contraction. It starts at the base of the hair and it goes to the tip. And as it goes, it expands and it squeezes those microbes out of pores back out into the soil. And then they reform their cell walls and they grow and begin to go out and get more nutrients. This is a sedge that'll illustrate that. The sedge grows in these limestone rock formations without soil, mostly without soil, but they do have lots of microbes. And this actually, below, the picture below shows root hair of the sedge. And you can see the bacteria inside that root hair. This is stained. You can see there at the outer area, just underneath the cell wall, at the periphery. And these actually are this is still here, but if you look at the living one, you can see it's motion. It's in motion, constant motion. This shows living root hair, and you can see these shadows going around. Those are bacteria. This is the way these root hairs move these microbes around. We don't think of root hairs as being action structures at all, but in fact they are. They're little machines that move these microbes around, and we think that's important because what that does is that will maximize the nutrient flow between the microbe and the plant, and the plant cell, the root hair cell. What happens is if it wasn't moving, you would get gradients, nutrient gradients that form, and that would slow nutrient flow. But because those gradients break down because of the movement, at maximum nutrient flow between the microbe and the plant. The other thing that happens when those microbes are moved around by the hairs is the microbes are broken apart, and they form these clusters of they're replicated, essentially cloned, and you can see like a cluster here that probably initiated from a single bacterium, a single cell that was moving around like that. This shows one of these hairs again. Here you can see the hairs going along in that cyclosis, and you can see the white little structures kind of jumbling around at the tip of that hair. That is the microbes. Those are the microbes in there, and that's pre-ejection. At some point there'll be a contraction, and they will be ejected out. And I'll show you images of that. Well, this actually is one of these hairs. You can see how fat they are. These hairs become fat, engorged with these microbes. So it's not just a few microbes. We're talking about lots of microbes. And this hair would be skinny if it weren't for all the microbes that are in it. Not all of them are staining, but you can see the microbes coming out of the tip there, or that came out, and you can see some of them are rods. They're reforming their rods. They cannot reform their rods so long as they're inside, because the plant is constantly bombarding them, controlling them with reactive oxygen and moving them around, keeping them off their toes, so to speak, so they can't come out. If we stop the movement, then the bacteria can reform their walls and then eat a hole, actually. They have enzymatic capacity to eat a hole out of the side of these. But so long as this root hair is working and functioning, they can't. They can't come out unless they're ejected out. This is tomato, and it's showing that ejection. You can see the expansion wave that starts at the base. And if you look at number 3 here, you can actually see 3 pores there with those microbes coming out those pores there. 3 little pores. Okay, so once they're ejected out, they will reform their cell walls and any flagella, whatever they need to swim out in the soil, and then go out and go about, be free living again, and then later attracted back to the root tip. So this central image just shows some of these hairs, and it's fluorescent microscopy, but you can see the bacteria that were squirted out of those hairs. And then actually when when you think of a hair, we've always learned that root hairs function to absorb nutrients, but in fact, for the rhizophagy cycle, they actually seem to function to move those microbes out into the rhizosphere, around the soil, out in the soil, away from the root, where there has not been absorption of nutrients, so that those microbes move them out so that in the soil they can then acquire more nutrients. It doesn't make any sense to put those microbes out right on the surface of the root where all the nutrients have been absorbed already. So what happens once this is actually clover, and these little dots in here actually show the protoplast phases. This is a yeast, actually. But it shows so yeasts also do the rhizophagy cycle. But you can see they're ejected onto the surface out from the tip of the hair, and you can see they're growing, actually. The youngest ones are near the tip, the oldest ones are away. What happens actually is once the plant ejects those microbes out of the hair, then it puts nutrients out, or puts nutrients out with the microbes. The flow of nutrients in these hairs is actually outward. So we think of nutrient as absorbing structures, but at least while these hairs are active, the nutrients are flowing out. What nutrients are we talking about? We're talking about carbohydrates. Carbohydrates are cheap for these plants because they're doing photosynthesis, so it's easy for them to feed these microbes. That just shows that again. You can see the microbes there, and you can see the protoplasts inside. And what happens to plants without these microbes? Okay, this is an experiment with rice. And here you can see in this experiment, all the microbes were removed from rice, and then we put it back on some of the rice plants. And this E plus here is with the microbes. This is a bacterial endophyte we put on. And you can see these seedlings got big. And this is without. The E minus means endophyte minus. And no microbes were put on. You can see those plants are repressed in growth. And when you look a little bit later, this is about a week later, you can see the ones with endophyte, you can see the plants are bigger. Roots appear more robust, but what especially is notable is they have all this soil adhering to them. Compared to without the microbes, okay, with the microbes, you have this close relationship of the seedling root to the soil. And that facilitates movement of nutrients into the plant. So these plants are better. Without those microbes, you don't have that close association. And so you have less nutrients. Those microbes are actually bringing nutrients in, but also there's an effect of exudation and so forth that's happening because those microbes are there. Okay, we've evaluated which nutrients are involved. And in one of these experiments, we used labeled nitrogen or isotopic nitrogen, and called nitrogen 15. And what we did was we put nitrogen 15 into proteins, and then we incorporated those proteins into this agarose, actually, which is gelatin substance. You can see here. And then we, in both cases, and then we put seeds that had their endophytes on them, their microbes on them, in one over here to the left, and those without microbes, or where we reduced the microbes, over here to the right. And what you can see is that where we had the microbes there, you could see, and we also stained this for reactive oxygen, so you can see all the purple to red, so you can see there's reactive oxygen there. But you can see the seedlings are also bigger. Okay, where we took the microbes away, you see little reactive oxygen. So you don't see much staining there. And the shoots are smaller there. But what's more important is when we measured the nitrogen 15 that went into the shoots. And this is with bacteria. You can see they went up to this high. This left bar, it absorbed this much of the nitrogen 15, whereas without the bacteria, and that's the middle one, it absorbed this much less. And actually, there's about a 30% difference. Okay, so between where we have bacteria and where the bacteria were removed. So we interpret that to mean that we're, in terms of nitrogen, the rhizophagy cycle microbes are worth about 30% of the nutrients that the plant absorbs, that in this particular experiment, this particular grass. We did other experiments, for example, this with winter wheat. And what we did was we isolated bacteria from the wheat, so we knew what the endophytic microbes were. And then we developed a procedure to clean up all the bacteria from the wheat and then grow it and then put the bacteria on one at a time. And so this actually shows one of those experiments. These are wheat plants where we grew without bacteria, and you can see they're what they are. But where we put the bacteria back on, you can see the plants are larger, and you can see the roots are much larger than over here to the left in the control without bacteria. So those bacteria really stimulated plant growth significantly. They were critical for plant growth. And then we measured nitrogen that went into or nutrients that went into those shoots of the wheat. And here, without bacteria, you can see all the way to the left, and no bacteria. It was a low level was some nitrogen that went in. It's not that no nitrogen went in. Some nitrogen went in. They acquired nitrogen. But where we put the bacteria on, and we had 3 different bacteria here, you can see they increased the nitrogen that went into those shoots of wheat by, again, about 30%. All of those bacteria were very similar. Phosphorus, you can see probably about 20% where we put the bacteria compared to no bacteria, again, about 20% increase. Potassium, for one of these bacteria, the first one, you can see it's like 3 times the amount of potassium. So potassium may be really important, and one of these bacteria are really good at carrying potassium into the plant. These other bacteria were not quite as good, but they still helped move potassium, and that's a point. And all bacteria are not the same. They carry nutrients differently, so they all have their characteristics of how they move nutrients. Here's calcium. You can see with the bacteria they did quite well. Sulfur, you can see sulfur with the bacteria they did better than the control. Manganese, you see increases with the bacteria there. Magnesium, zinc, iron, boron, both macro and micronutrients were impacted, and it depends on which are. Can you add commercial nutrients? Yes, you can. You can add commercial nutrients, and it works perfectly fine. And we've done experiments there. One experiment, what my student did, actually, this is with the celery without microbes here, and then we took a commercial microbe, we put it on the celery, and now you can see the root hairs there. Okay, but you have to always test these microbes. And this actually, from the celery, you can see the microbes actually in the root hairs there. Always test your products, do experiments. Don't take anyone's word that something's going to work. Be sure you do tests. Okay, and these microbes will actually improve healthful components in plants. Like, for example, we did a carrot experiment, and where we took some different bacteria, several bacteria from different plants, and we put them on carrots, and then we measured actually carotenes in the plant. And just to get to that, okay, this is 3 different bacteria. The control to the left shows the amount of carotene that we measured without bacteria. Then where we put bacteria, we see one bacterium didn't do anything, but another bacterium, you can see it looks better than double the amount of carotenes that were produced. And the third one gave a little bit more. Okay, so different bacteria will do differently. Okay, this is beta carotenes, basically the same effect, no difference there. Okay, so there's and this is basically my conclusion slide, but these endophytes in the rhizophagy cycle will actually carry nutrients into plants, and the plants will extract those nutrients from the microbes. That's a benefit to the plant. But they also, because the plants are oxidizing them with superoxide, reactive oxygen, essentially, the plants are producing reactive oxygen, then the plants also are upregulating their antioxidants and their oxidative stress genes, their antioxidants and other chemicals that protect them from their own reactive oxygen. And that actually makes the plants more resistant to oxidative stresses like salt, or heat stress, or whatever, it could be heavy metal stress in the soil, whatever the stress is that is oxidative, and most stresses are oxidative. So when plants get their nutrients from the rhizophagy cycle, they become more hardy, they become hardier and more stress resistant than if you're just feeding them with fertilizers. And finally, many of these bacteria that participate in the rhizophagy cycle will also go out and they'll colonize soil fungi, and they will make those soil fungi, those pathogens that are in the soil, they will make them avirulent. They will colonize them on their surface, and they'll produce things like, for example, those lipopeptides that I talked about that make the membranes leak, and they get the energy, essentially, get the sugars and other nutrients that leak out of those fungi, and those fungi become avirulent, and they no longer can cause disease. They don't have the oomph to cause disease. So, take-home message here, preserve your microbes on seeds. Don't do anything that would hurt those microbes. Manage soils so that they're living soils. You have lots of microbial activity in those soils. The more microbial activity, the better. You can use biostimulant microbes. They help. They do help. We've done experiments that show they help. They do participate in the rhizophagy cycle, but you need to test to make sure that they actually are helping in your system, helping your plants. And finally, endophytic microbes can improve quality in crops. These are some rhizophagy articles that you could get that you could find One is called rhizophagy cycle, an oxidative process in plants for nutrient extraction from symbiotic microbes. It's in microorganisms. If you just Google rhizophagy cycle and microorganisms, this will pop up. There's another review article about endophytic microbes that is in actually Pest Management Science from 2019. Also open access, you can get to that. And questions now. I hope I haven't gone too long.
All right. No, I think this is a good timing here. We've got about 20 minutes left to get some more questions in. So thank you for that, Dr. White. We had a lot of people's comments saying, wow, this is amazing information, and people really liked the videos and the photos. So thanks for that.